So the final part of respiration then is three little bitsy bits involving RQs, alternative substrates, and the respirometer. So if we start here with alternative respiratory substrates. Most of the time that we've talked about respiration, we've always been looking at the respiration of glucose, which involves these steps: glycolysis, breaking down the glucose into pyruvate, then the link reaction, then Krebs cycle. Along the way, we get loads of hydrogen atoms released, which get transported by the NAD or FAD to the electron transport chain. But in reality, we don't always have glucose available. What can happen sometimes is we need to use fats or proteins for respiration and as an energy source. So if we look at fats and proteins here, we're basically going to see where they enter these different stages. So if we take fat first of all, it gets broken down into glycerol, and then the glycerol gets converted into a triose sugar, and then it basically enters respiration at this stage. So fat will get broken down into glycerol, and then it'll enter in this way, and it'll behave just like a triose sugar. But obviously we know the fats are made up of glycerol and fatty acids, so the fatty acids will enter the chain here. So the fatty acids can be long hydrocarbon chains like this, where each part will contain carbons bonded to hydrogens. Now what happens is the big long fatty acid chain gets chopped up into chunks of 2 carbon molecules, and then the 2 carbon molecules get fed in here and join up with acetyl coenzyme A. Now, it's important to say here that for fat to get broken down, it's a hydrolysis reaction. So when fats are hydrolyzed into glycerol and fatty acids, the glycerol and the fatty acids enter at different stages. It's also an important point here to say that if we're looking at carbohydrates, they basically only form 16 kilojoules per gram of energy, whereas fats produce double that at 39 kilojoules per gram. So this basically means that if I were to eat 100 grams of glucose, it would provide me with 1,600 kilojoules of energy through respiration. Whereas if I ate 100 grams of fat, it would be 3,900 kilojoules of energy per 100 grams. So clearly fats contain much more energy than carbohydrates. The next respiratory substance that we can use then is protein. Now, proteins are only going to be used for respiration whenever we are near starvation. So your body would choose carbohydrates first, then fat second, and then proteins only whenever you're near starvation. So the protein then is hydrolyzed into amino acids, and then the amino acids get deaminated, which basically means that they have their amino group removed or amine group removed, and then they'll get fed into different stages in respiration. Now, because amino acids are so varied, that's why they can enter at different stages. So once the amino acids have been deaminated, they can either enter in at the stage with pyruvate or oxaloacetate or oxoglutarate. But as I say, our body will prefer to use glucose as a respiratory substrate, then fats, and then proteins, but only in starvation conditions. Now, what you might have noticed here is that glucose, fatty acids, and even amino acids can enter the chain, and they all end up at acetyl coenzyme A. Because a lot of them end up with acetyl coenzyme A at some point during respiration, it's sometimes known as the hub of respiratory pathways. And that's basically because no matter if it's glucose, fats, or proteins, at some point their respiration will involve the production of acetyl coenzyme A. Now the next little part is about respiratory quotients or RQs. So the respiratory quotient is a measure of the ratio of carbon dioxide evolved by an organism to the oxygen consumed over a certain period of time. And there's actually two ways of calculating this. So RQ can either be calculated by taking the volume of carbon dioxide released and dividing it by the volume of oxygen consumed, or you can use a balanced equation and you could work it out from the number of molecules of carbon dioxide released divided by the number of molecules of oxygen consumed. Now, the whole reason why you would do this or work this out is because if you work out the RQ value for a certain organism, it will tell you what sort of substance is being oxidized or what substrate is being used in respiration. Theoretical respiratory quotients for the complex oxidation of carbohydrate, fat and protein can be worked out from the chemical equations, which is basically talking about that equation there. So if we split this in half here, we can show you how you would do this using the balanced equation for both glucose and palmitic acid. So first up is glucose. Now, we should know that the equation for the respiration of glucose is C6H12O6 plus 6O2 produces 6CO2 plus 6H2O plus 38 ATP. But the ATP is not the important thing here. What is important is the number of molecules of carbon dioxide released, so 6 CO2 divided by the number of molecules of oxygen consumed, which was 6, which equals 1. So our RQ for glucose equals 1. Now, just to keep track of where we got those, this was the number of molecules of carbon dioxide released, and this was the number of molecules of oxygen consumed. The next one then is palmitic acid, which, don't worry, you don't need to learn this one, it's simply just here for an example. Plus 23O2 produces 16CO2 plus 16H2O plus energy. So we take the number of molecules of carbon dioxide released, which is 16, divided by the number of molecules of oxygen consumed. So again, let's just keep track of where we're getting these numbers, which gives us a value of 0.7. So the RQ value for palmitic acid is 0.7. Now, these were very carefully selected ones. Okay, the reason for that is because carbohydrates, fats, and proteins have typical RQ values. For we've got substance, so carbohydrate, fats, proteins, and then typical RQ for each one. So for carbohydrates, it's 1. For fats or lipids, it's 0.7, and for proteins, it's 0.9. Now, as we know, proteins are a really varied group of compounds. So the composition of proteins is too varied for them to give the same RQ, but most have a value around 0.9. Resting animals have an RQ of between 0.8 and 0.9, and the RQ for humans is generally about 0.85. Now, 0.85, this doesn't really fit in with these values that we've just looked at, and there's a very good reason for that. So why do organisms rarely have an RQ of 0.7, 0.9 or 1? Well, for humans, it's basically 0.585 because they use a combination of carbohydrates and fats usually. So why do organisms rarely have an RQ of exactly? It's because most use a combination of respiratory substrates, e.g. humans use mainly glucose and fats. Now, if you were to take 0.7 plus 1 and then divide it by 2, you would get 0.85. So it's basically an average of the RQ of glucose and fat. Now, the RQ can also indicate a type of metabolism occurring. What might a high RQ above 1 indicate? Well, that basically means that there's more CO2 released than oxygen taken in. So it means that an aerobic respiration is taking place. Now, if you think about it, it makes sense because the equation for anaerobic respiration in mammals would be glucose, which releases carbon dioxide plus lactate. So looking back at how you actually calculate an RQ, oxygen's nowhere to be found here. So obviously, to work out the RQ for this, it would have to be done using volumes. And to calculate the volumes, that's where the respirometer comes into it. Now, I should add a wee note here that when you're exercising, yeah, okay, anaerobic respiration will take place, but it's not like a switch where you go from aerobic to anaerobic. If you were working really, really hard, like during a run, and you could feel lactic acid building up in your muscles, you would still be respiring aerobically. So it's important to say here that if you've got an RQ value calculated of above 1, that anaerobic respiration is taking place, but it's at the same time as aerobic respiration. Because as I say, it's not like it's a switch. You don't just all of a sudden stop aerobically respiration and start anaerobically respiring. Both will take place at the same time. And I suppose it's important to mention here that because aerobic respiration will still be taking place, that if you get a value of more than 1, that's likely to indicate that it's a plant or a fungus which is anaerobically respiring. Now, the simple respirometer. Everybody's favourite. This can be used to calculate respiratory quotients, and also it allows you to calculate values for metabolic rate, which basically means how much oxygen is being consumed per unit time, per unit mass. So for example, I could calculate the metabolic rate of these little larvae, and I could maybe calculate it in terms of oxygen consumed per hour per gram of larvae. Now, this is a small respirometer, which we've seen before. And the process is fairly simple. So let's have a little reminder about how it works. So, the respirometer here then is used to calculate oxygen consumption and carbon dioxide production. You basically have a solution at the bottom which contains either potassium hydroxide or water. You have a living material, so you might want to have like larvae, wood lice, even peas, something like that. You have a little capillary tube, a little millimeter scale, and then a coloured bead of liquid. And basically, the premise is that you measure how far this coloured bead of liquid travels in a certain period of time. So to use the respirometer, it relies on pressure changes. Any increase in pressure in the tube makes the bubble move away from the tube. So if the pressure in here increases, the bubble is going to move that way. Any decrease in pressure, the bubble will move that way. And if there's no change in pressure, the bubble doesn't move. Now, this basically just shows you what happens in order for you to calculate oxygen uptake. So, here we have potassium hydroxide in here. Here we have our living material. Now, what's going to happen is oxygen's taken in, the animal respires, and releases carbon dioxide. The carbon dioxide gets absorbed by the potassium hydroxide, so the volume of air in the tube decreases. That has the effect of decreasing the pressure, and the bubble moves towards the test tube. The distance the drop moves shows you the volume of oxygen consumed. So initially, the key points are this. The respirometer is set up with potassium hydroxide. The animal respires, it releases carbon dioxide. Carbon dioxide is absorbed by the potassium hydroxide. The pressure drops and the bubble moves towards the tube. And the distance the drop moves is equal to the volume of oxygen consumed. To calculate carbon dioxide production, you reset the apparatus and replace the potassium hydroxide with water. This time, any carbon dioxide which is produced is not absorbed. Now, because we already know how much oxygen is used, we can use this to work out how much carbon dioxide is produced. So this is the key point. To measure oxygen uptake, we need to have potassium hydroxide in the tube, and the organism respires, releases CO2, which gets absorbed by the KOH. So the pressure decreases and bubble moves towards the tube. The distance the bubble moves equals oxygen consumption. Next, the carbon dioxide production. So what you would have to do is reset apparatus, use H2O instead of KOH, and depending on the bubble movement, work out carbon dioxide production. Right, now calculating carbon dioxide production is the difficult part, so let's have a look at it. After you've done it with potassium hydroxide, you reset the apparatus and put in water. Now the water is used to measure carbon dioxide production, so you've got three outcomes here. Either the liquid doesn't move, the liquid moves away from the tube, so the pressure in the tube is increased, or the liquid moves towards the tube. Now, depending on the outcome and how far each one moves, this tells you how much carbon dioxide was produced. So if the liquid doesn't move, that means that the volume of oxygen consumed equals the volume of carbon dioxide released. So let's say, for example, you set it up the first time with potassium hydroxide and the bead moved 20 millimeters. You then set it up and repeat it with water, and this time it doesn't move. Well, that means that it took in 20 millimeters of oxygen, but also released 20 millimeters of carbon dioxide because there was no movement in the bubble. The second scenario is that the liquid moves away from the tube. Now, this will actually mean that the volume of carbon dioxide produced is greater than the volume of oxygen consumed. And the reason for that is, if this is an enclosed space in here, and there's more carbon dioxide being released, it's going to increase the volume of air in here, increase the pressure, and push the bubble that way, away from the tube. The final scenario then is, if the liquid moves towards the tube, that means that the volume of oxygen consumed is greater than the volume of carbon dioxide released. So basically what's happening in that scenario is that the living material is respiring, but it's taking in more oxygen than it's releasing, so the pressure drops and the bubble moves towards the tube. Now, in order to calculate the RQ for respiring organisms, what you need to do is first calculate oxygen consumption. Second of all, calculate carbon dioxide production. And then you need to do volume of CO2 produced divided by volume of oxygen consumed, and you'll get a value for it. Now, just while we're here, we may as well talk about controlled variables. So it's important that when you're doing this experiment, you use the same apparatus for calculating oxygen uptake and carbon dioxide production. You have to use the same organisms. So factors such as age and metabolic rate are controlled. So for example, in this one, if I was using wood lice, I would try and use the same wood lice to calculate both oxygen consumption and carbon dioxide production. You would also try and control the temperature. So to do that, you place this whole apparatus in a water bath, because remember, respiration is exothermic, so the temperature will actually increase, and it'll maybe even be exacerbated because you've got these organisms in a little test tube, so it's more likely that the temperature will rise. So you place it in a water bath and that controls the temperature. Then you need to make sure that you use the same time period for oxygen and carbon dioxide calculations. And this diagram here just shows how a respirometer might be carried out in a lab. So it's within a water bath. We have one test tube here, another test tube just in behind, only this one has a U-bend. Now, the last little part of this topic then is to use redox indicators. Now, this basically just shows you what's happening at different stages. So at several stages in respiration, hydrogen atoms are removed from molecules in reactions by dehydrogenase enzymes. So dehydrogenase enzymes catalyze these reactions, and the process is described as dehydrogenation. The hydrogen atoms are taken up by NAD or FAD, forming reduced NAD or reduced FAD. It's possible to use redox indicators to demonstrate dehydrogenation taking place. These indicators, as opposed to NAD or FAD, take up hydrogen and change colour when they become reduced. By adding a redox indicator to suspensions containing living material, for example yeast, it's possible to demonstrate dehydrogenase activity or to compare activity in different conditions. So here we have some redox indicators, and we've got methylene blue. When it's oxidized, it's blue, but when it gains hydrogen and becomes reduced, it changes to colourless. DC-PIP will go from blue to colourless as well, and TTC will go from colourless to pink. Now, by far, the most common one that you'll get asked about is methylene blue. So, it will be blue in its oxidized form, but when it picks up a hydrogen from respiration, it will change to colourless, and that's in its reduced form. So remember, reduction is the gain of hydrogen. So in this case, the blue methylene blue gains hydrogen, becomes reduced, and changes to colourless. The only way to change it back then is to remove the hydrogen or add oxygen. Now, by far the most common process that you would be looking at is this, showing that respiration is taking place, indicated by the blue indicator changing to colour. Now, here's an example, albeit it's very, very straightforward compared to the questions you might get. So comparing respiration rates of living and boiled peas. Now, the difference is the living ones can respire, whereas the boiled peas are probably going to be dead and there's going to be no respiration because all the enzymes would be denatured. So here's what you would do. You would crush soaked peas into a paste in a pestle and mortar and place it in a boiling tube. Do the same thing with boiled peas. So you basically have 2 boiling tubes with a crushed up paste of living peas and dead peas. You add the methylene blue to the boiling tube. So here we are adding our methylene blue. And then you place in a water bath at 35 degrees C and observe the colour changes. Now the colour changes that you'll get would be that the one in the living tube and that is the end of this section.